All right, so we'll jump into Anandan's presentation. So Anandan's was part of Frontier some years ago. Did a project with Michael Hornberger. I can't remember when it was. I think 2013, 2012,
Yeah, 2012. Yeah,
2012. And then um, decided to move to Canberra for some unclear reason. Now, did his PhD and looking at different variables contributing to brain health, including sex differences, which is really interesting. So he's a lecturer at University of Canberra, and yeah, so he's going to tell us about big data and how to use big data in looking at risk factors for brain health. So Anandan, welcome, and floor is yours.
thanks, Olivier. Thanks, everyone. Thanks for the invitation to speak today about research. And it's lovely to be back. I've titled this talk "From Frontier to Frontier: Investigating Brain Health Risk Factors Using Big Data Analysis." And I thought it would only be appropriate if I started, I guess, on the back of what Olivier said in regards to where I started this journey and interaction with Frontier. So this is photo of me, and many years ago. I first connected with Michael Hornberger. At the time, you may or may not know, and still in touch with him in regards to. I was a first year undergraduate student, and I was interested in science, interested in neuroscience, but didn't really know much more than the basic units I was doing. And so I was keen to get my hands involved in research. Michael, for some reason, was happy to have me involved in what he was doing, and I worked on a small project with him. Learned a lot of experience in regards to how research is conducted and whatnot, and it led to a paper publication, which was what I didn't realize at the time was very valuable to me in future. But at the time, I was just learning as much as I could, and that internship led to several others as well, including ones with Tim Carroll. And so I did some animal research, and then realized that the research I did with Michael was of more interest, and connected again with Olivier, who Michael put me in touch with. And Olivier, I was working with in my final years of my undergraduate degree, part volunteer internship, part of it informed some of my coursework, which was really nice to see. And there's a lot of value. I, again, learned a lot of the hands-on skills in research, but also for a period of time, I was situated just outside Olivier's office. And so I got sort of an informal training in terms of the day-to-days of academia, in terms of research and the challenges that emerge and how to adapt and respond to those challenges. And I didn't realize at the time how valuable that time would be for me, particularly at this stage of my career. So I'd finished my undergraduate degree with the intention of doing honors, but got a job as a science communicator at Neuroscience Research Australia. And so I worked there for a very short stint of time in science communication. But in my undergraduate degree, I taught at a tutoring college. And in this tutoring college that I taught at, there was a classroom, 15 students. There was a whiteboard, a textbook, so it felt like a typical teaching experience. And I really loved teaching. It was really valuable and moments in my mind when it comes to how my teaching journey started. And so I had to give that job up to do the science communication. I realized very quickly that teaching is what I wanted to do. So I ended up doing the master's in teaching in secondary education. So I completed that soon after my undergraduate. And from there, realized that university is probably the context in which I wanted to teach in. So that led me to my PhD. And so I went to ANU, as Olivier said, in Canberra, to do my PhD with Nicholas Shabern. I should note that these photos are timestamped. So this is a photo of Olivier and me in this time window. And I learned really, I got a good training in many of the skills that I use in my research career now in regards to R, statistics, Git, Linux, neuroimaging analysis, big data analyses. I was very lucky. It was, the PhD was a challenging time. It was difficult, but it was incredibly rewarding. And I think in part it's because of the number of skills that I learned as part of that PhD journey. So that was really useful. And as I was wrapping up that PhD, I got a job at the University of Canberra as a lecturer. And so I've been working there ever since as a lecturer. I'm in the Faculty of Health in the discipline of psychology. And so looking at these photos and seeing the changes over time, I realized that I didn't know if I was really qualified to be an aging researcher. I mean, when I look at Olivier across these photos, see, it's true. If he hasn't changed, he's only looked younger as time has gone on. And so, Olivier clearly possesses the secrets to aging. But the more I thought about it, I thought, perhaps I do belong in the aging space because I've been ravaged by the time of aging. I've experienced aging so immensely. I have an intimate knowledge of how aging is and how it occurs. So I guess maybe it is an appropriate research space for me. This is my website. I'm happy to share these slides afterwards, by the way, but for future reference, anything that's read or hyperlinked in my slides is hyperlinked. So you're welcome to click that. It'll take you to a source or a paper or whatnot. And so largely research, teaching are core components of what I do. It's a research and teaching role here at the university. And I also take on a role in regards to the emerging generative AI in tertiary education space. And so largely my role within the faculty is to upskill and help others learn how to utilize these technologies to improve their teaching, research, and professional practice. But that's sort of a broad summary of my background and what led me to here today. And I thought it'd be nice to reconnect with Olivier and the Frontier Lab. And really today is sort of like an olive branch to give you a glimpse of what I've done, the research I do, and the questions that sort of motivate me to do the research I do. And perhaps that'll lead if you find that you have similar interests or similar research projects that we can collaborate on, because I think a challenge I have here in the university is the number of researchers specifically in my field, or neuroscientists in my field, is very limited. And so largely to achieve the impact that I'm hoping to achieve, it really relies heavily on collaborations with other researchers. And I've been successful with that in terms of continuing to work with Michael, who's located over in University of East Anglia, and I've also developed connections in Lausanne in Switzerland. But I'm open to finding the right fits as well to ensure that we can pursue research, because in part, some of the fun of research comes from really nice collaborations as well. So I thought I'd start this talk with an overview of the questions that really motivate me in research, the ones that I think a lot about, and the ones that drive the projects that I commit my time and effort to pursuing. And so fundamentally, the first question is, what are the causes of dementia? And so that leads to an investigation of the mechanisms that contribute to aging and the pathology of dementia. And there's different ideologies when it comes to dementia and aging. Some people think that dementia is completely separate to aging, isn't a normal part of the aging process. Others think that there's some degree of overlap, and others believe that it sits on a continuum. And I think for me, these discussions about where dementia fits in this space or whether it doesn't fit is in some ways confounded by lifespan. And when I ask myself the question, if everyone in the world was to live to 300 years of age, would they develop dementia or some pathology similar to dementia? And I think the answer, I say, is for most people, yes. And if not at 300, it would be earlier. And I think to some degree that informs the ways in which I think. Like, of course, there's a theory of evolution would suggest that maybe there'd be some population within there that would be resistant to developing dementia. But I think largely from pretty much 99% of people, you'd expect that the accumulation of pathology over time and the degradation of these mechanisms that drive aging would contribute to the development of dementia. So that's really where my thinking and perspective comes from, and informs much of how I view the relationship between risk factors and dementia, and also the life course model that I adopt when I think about these issues, which I'll touch on in a bit. And so that's more the theory and my interest in terms of the mechanisms that drive the pathology of dementia, but also I focus on genetics, environmental and lifestyle factors, cardiometabolic factors, which for me, I think is one of the major contributors to dementia risk, and in regards to particularly the overlap that we see in vascular dementia and Alzheimer's disease. I think the notion that what's good for the heart is good for the brain it rings true, and really led to a lot of the focus of the research you'll see that I've done focuses on cardiometabolic factors in addition to sex-specific factors. Now, a second sort of question that motivates what I do is how can we effectively utilize available resources to reduce dementia risk? Because I think we need to understand the risk factors, their contributions, the magnitudes of their effects, and then use this understanding to inform the population about how they can best reduce their dementia risk effectively, so that they either delay the onset or progression of dementia, so that it extends beyond, hopefully, the lifespan. And so I think in part this, for me, leads to two fundamental components. One is ensuring that there's accessible measures of brain health, and then also that these measures of brain health accurately predict dementia risk. Largely, if we want to understand how a person's brain is functioning, this relies on neuroimaging and neurocognitive assessment, which typically are reserved for those who have flagged some issue in regards to their cognitive capabilities, or if they've had some head trauma or suspected dementia query. And so I think the two elements of this. First, I think there needs to be the development of an accessible brain measure that allows for people in the public to get a sense of how their brain health is performing. And I think one possible measure, although there's several, one possible one is brain age gap, which looks at it can utilize either structural imaging or multimodal images to inform an brain age estimation. And the difference between the brain age estimation and an individual's chronological age gives some information about how their brain health is. And so if the brain age is higher than the chronological age, then you would think that the age of brain aging is accelerating. And if it's lower, then the brain aging is positive. And so the accurate risk prediction, I think, is a major challenge in the field when it comes to dementia risk, and being able to ensure that the models that we produce accurately predict time to conversion for dementia and predicting the risk of developing dementia. And this is where I think in part and I'll touch on this later in the presentation, in regards to developing prediction models across the life course are really important, not just looking at risk in late life, but also midlife and arguably early life as well. But particularly midlife poses a time for where intervention and strategies can be put in place to reduce risk of developing dementia. And I think when it comes to these dementia risk models, what's really important is to ensure that we quantify dementia risk at a level which is meaningful to an individual. I think in many times when I'm involved in this research space and we look at these models and how they predict dementia risk, the question I often ask is, how is this meaningful to someone off the street? More above and beyond just telling them what we already know are generic factors to improve brain health: exercise, eat a nutrient-rich, varied diet, don't smoke, don't drink excessively. How does a dementia risk prediction tool provide information to an individual above and beyond these sort of broad statements? And I think that's where the power of these tools needs to be focused on, in terms of ensuring that we can have these targeted interventions that improve health for the individual, considering their individual history. And that is a theme that sort of emerges in my research as well. And then the third sort of facet of the motivations of why I do what I do is understanding how we can effectively engage the public in scientific research. So, as I mentioned, I was a science communicator for a short stint of time, and it's something that is a passion I've connected with throughout my career. And in part, it's because it allows individuals to make informed decisions about their health. It informs critical thinking and debate, and I think an informed public is a good thing for society. And so this sits with informing policymakers and health professionals and the community and those with lived experiences. Now, arguably, I don't do all of these things very well. I'm still broadening my reach to ensure that I'm connecting with policymakers and health professionals. And I'm part of the Centre for Aging Research and Translation at the University of Canberra. And there's some researchers here within the centre who do an amazing and inspiring job in regards to working with people who suffer from dementia and those with lived experiences in the community. And I know that Frontier is an exemplar of this as well in this space, but this is certainly something I'm hoping to improve upon myself. But I think what I do excel in is the teaching and the science communication. So I view teaching as a pathway to help teach students this scientific thinking, being able to think critically about information that's presented. And this, in my view, allows me to have impact that extends in this research space if they pursue a research career, or into the public if they go elsewhere as well. And this largely is my interest and why I put time and effort towards science communication and teaching as well. And so as a background in regards to dementia, it's a growing issue and will continue to grow in terms of the incidence of dementia. It's predicted in 2050 global incidences at over 130 million cases. But this picture doesn't really show the differences that we see by sex. This is just sort of the whole population. And when we look at sex differences in dementia incidence, we can see that it increases in both men and women, unsurprisingly, but women seem to be disproportionately affected by dementia. And this is clear as well when we look at risk factors and the leading causes of death in Australians. And we can see that for men, coronary heart disease or cardiovascular issues are one of the leading causes of death in men, followed closely by dementia. But in women, dementia is the leading cause of death, and then followed by these cardiovascular diseases. And so, in part, much of the literature has put this down to the age differences between men and women. The life expectancy for Australian women is 85.3 years, and men it's 81.2. So the fact that women live longer and age is a big predictor of dementia risk seems to inform our thinking about how well it makes sense as to why women have this increased risk in dementia. And this is consistent not just in Australia, but like with global trends. We see that women on average live longer in pretty much all, if not most countries, although the differences vary depending on the countries. But on average, it's about a 5-year difference between men and women. And so an interesting paper came out in 2019 that looked at age standardized global prevalence in females, and it's for dementia risk. And it found that it was 1.17 times the age standardized prevalence in males in 2016. And so this shows that, yes, age is a big factor. It contributes to the difference that we see in dementia prevalence between sexes, but it doesn't explain the entire story. And so there's other evidence that seems to indicate sex-specific effects when we look at the brain. For example, whilst the frequency of apolipoprotein E, major risk factor for dementia, well, E4 variant, it doesn't differ by sex. A meta-analysis indicated that women with the E3, E4 genotype had an increased risk for Alzheimer's disease compared with men between the ages of 65 and 75. So we see these sex-specific differences that occur. And when we look at things like cognitive decline, particularly in those with mild cognitive impairment, we see in a longitudinal study using the ADNI cohort, Alzheimer's Disease Neuroimaging Initiative, they found that for those with mild cognitive impairment, the actual cognitive decline or rate of decline was faster in women than men. And these models were adjusted for ApoE, education, age, baseline cognition as measured by the MMSE. So the usual suspects that you'd suspect. So essentially, we're seeing these sex differences emerge. And the question, I think, becomes, where do they fit within the scope of lifespan? And what are the potential contributions or factors that contribute to the differences we see in sex? And this is an amazing study that done recently that looks at developing a brain chart for the human lifespan. If you've had a child, or people who've had kids, you may notice that the weight and height of the children are measured against these charts that are normative data. And we see this also in neurocognitive assessments, where we have these norms about how people should be performing on different assessments. And And these researchers realized there's a gap in terms of structural brain imaging in regards to having established norms of how the brain changes over time. And what they did was incredible. They aggregated over 120,000 MRI scans from across 100 different studies. And they looked at over 100,000 participants between the ages of 115 days post-conception to 100 years of age. And you can see all sorts of things there. I'm not sure if you can see my cursor on the screen as well, but over here you can see the UK Biobank study, which is something I've worked extensively in, which has participants aged 45 to 60. And in addition to that, all these other neuroimaging data sets are available. And what we see here, apologies for the amount of information here, but I think what I want to draw your attention to is the fact that when we look at the rate of decline in the structural volumes over time, we see that this occurs certainly in late adulthood and middle adulthood, but to some degree it can occur in young adulthood and adolescence, particularly when looking at gray matter changes that occur. And so this gives some indication that the changes that we see in the brain occur over the lifespan, and perhaps it's the accumulation of pathology over time that contributes to these neurodegenerative conditions, including dementia. And here, again, I want to, this is quite a nice picture that I've painted, but I want to point out the realities of the data. And when you look at the top row of data, I want to draw your attention to the inter and intra variability that we see for sex when we look at these trajectories in brain changes. And so we can see that there's differences between men and women in terms of these trajectories, but also within the groups of women and men, there's also quite a lot of variability. And this speaks to different exposures to risk factors, perhaps, that contribute to changes in the variability, perhaps also genetic predisposition. There's many factors that contribute to it. And I think it's important to constantly keep this in your mind when you consider research, as the variability that we see, particularly when the estimates are often averages or group averages rather than capturing this individual variability. And an interesting, coming back to the idea of normative data, in the UK Biobank, there was a study done recently that looked at plotting the brain changes that we see across individuals in the data set, which is 50 to 75 here. And what we see, which is of interest, is the change in hippocampal volume. So the plot on the top left, you can see that there's a steady rate of decline in males, but in females, there seems to be some acceleration that occurs around 63 to 67 years of age. And so the question becomes, what are the factors that may contribute to this change in rate of change over time? And so, and in both men and women, there were increases in the rate of hippocampal volume loss relative to the rest of the gray matter, around ages 67 for women and 63 for men, which is this plot here. But the hippocampus, the authors posited that they may have particular vulnerabilities to aging, as effects weren't detected in neighboring regions, including parahippocampal gyrus and the temporal gyrus. So interesting to see the sex effects emerge. And so, but when we look at other markers of clinical disease stage and biomarkers, we can see that, well, for sex differences in regards to amyloid beta and tau burden, the evidence, I could present evidence that shows increasing tauopathy in women compared to men. There's certainly research out there, but the reality is when you scan the literature is it's very limited and requires further replication and to really understand the contributions of this. And these are very difficult biomarkers to assess as well. And one of the key factors that contributes to a lack of a clear understanding in this space in regards to these biomarkers is that statistical models, they often adjust for sex, but they don't conduct sensitivity analyses that stratifies analyses by sex or investigate potential sex interactions. So whilst many in the neuroimaging space, when many people control for sex as a factor, many people don't do subgroup analyses or sensitivity analyses that look at within sex differences. And an example of this is a meta-analysis conducted on sex differences in brain reserve, as defined by the authors as IQ, education, occupation, cognitive activity, multilingualism, socioeconomic status, physical activity, social support, or marital status. So all of these outcome measures, out of all of these, they identified 16 studies that could be included, that included an analysis of sex. And so this sort of speaks to the limited research there is in regards to sex differences for brain health. And so what are possible reasons for these potential sex differences? Well, I think there's some very plausible reasons that emerge, including historical inequities resulting in disproportionate access to education and occupational opportunities, which we know contribute to brain reserve. There's also potential selective sex bias in men. So with those 65 years and older, they've lived through midlife to some degree, and so they're likely to have a healthier cardiovascular profile, and therefore maybe they're less likely to develop Alzheimer's disease. That's one possibility. The other possibilities are interactive effects between sex and sex-specific factors and genes. So there may be some interactive effects that are occurring that are contributing to the differences we see, and also there are unique neuroendocrinological processes that occur in women, including menarche, menstruation, pregnancy, and menopause. And so I was interested for much of my early research and PhD in regards to the contribution of menopause and the changes that occur around menopause and those changes and their relationship to the brain. So menopause, as a brief overview, it comes from Greek words meno, which means month, and pause, which means stop, thus indicating the end of monthly cycles or menstruation. And it's really defined as the absence of menstruation for 12 months, for at least 12 months. And so it's only defined retrospectively. Now, it's historically been understudied in the context of aging and neuroimaging literature. So over a period of 23 years, peer-reviewed neuroimaging articles which focus on menopause accounted for approximately 2% of the aging literature. Affects almost 50% of the population and in regards to menopause as a process. And so the average age of menopause lies between 46 and 52 years of age, with the average age around 48. And typically this equates to around 40% of the life of a woman is in postmenopause, so in a postmenopausal state. And so here's the current conventions in regards to defining menopause. They're called the Stages of Reproductive Aging workshop, which was developed this nomenclature for defining menopause. And in part of my systematic review, I sort of teased apart how well these criteria were adhered to by others and proposed some suggestions for improvement. For example, premenopause is a term that's widely used, but is not represented in this diagram. But in any case, what I want to draw your attention to is the endocrinological changes and physiological changes that occur around the menstrual cycle and across these stages of reproductive aging in women. And particularly how when we look at things like where symptomology occurs around perimenopause and postmenopause, which overlaps to some degree with perimenopause because of that year gap. And so my interest was in what were the biological and physiological changes around menopause, with a particular focus on those changes that were of largest relevance to brain health. So this, for me, led to an investigation of cardiometabolic factors. And an interesting question that's really difficult in this space is understanding or delineating how much of the effects that you can see attributable to menopause versus attributable to aging, because menopause occurs concurrently with aging, right? So they're highly correlated, and so trying to tease apart these effects requires some complex statistical analyses, and which I'll walk you through as well. So this is a study, a systematic review I conducted, which looked at fat mass changes during menopause. There's quite a lot of debate in the literature in regards to I felt like there was in regards to how much of the changes in fat mass were attributable to aging and or menopause, with people sitting on either sides of the camp. And I think in part there were many factors that contributed to this, in addition to the difficulties I've raised already, but also the inadequate consideration of different measures of fat mass. And so I conducted the systematic review, which it consisted of 201 cross-sectional studies and 11 longitudinal studies of over 1 million women in total for the cross-sectional analyses. And it looked at all sorts of fat mass measures. If you name a fat mass measure, I'm quite confident that it was investigated in this study. Body mass index, total fat, visceral fat, waist circumference, waist to hip ratio, all sorts of fat mass measures, because we were interested in knowing precisely quantifying the changes that we see. And here you can see the differences in trunk fat percentage between, so trunk fat being the central fat measured by DEXA, dual X-ray absorptiometry imaging. And we see that difference between pre and postmenopausal women was around 5.5% difference there. And so the key findings that emerged from this the systematic review was that, yeah, fat mass increased between pre and postmenopausal women. So postmenopausal women had, on average, higher levels of fat mass than premenopausal women. And this was largely attributable to aging. So when we included age and meta-regression, it accounted for a significant amount of the unexplained variance. And when we looked at longitudinal trajectories, or studies that have mapped longitudinal trajectories for changes in fat mass in women, they matched, if not in some cases closely, if not exactly, to the changes that we saw in our study across menopause. And so there was no really detectable change in the slope across the menopausal period. And there was no interaction for age and menopausal status for fat mass. So the association between age and fat mass didn't depend on menopausal status. However, we found something really interesting. So that was in relation to total fat mass, but we found something really interesting when it came to fat mass distribution. With increasing, of course, there was increasing central fat, but we found a decrease in leg fat. And so it was posited that possibly hormonal shifts around menopause may have contributed to the enhanced central fat deposition. So we did subgroup analyses in women who had used hormone replacement therapy. We included them in the analyses, and then we excluded them, and we saw what the difference was in the results. And what we found was that when we included women who used hormone replacement therapy, there was a significant increase in body fat percentage and decrease in trunk fat percentage, so that central fat, which was suggestive to us of a possible protective role of hormone replacement therapy in preventing or reducing trunk fat deposition, but not in preventing overall fat mass gain. Now, you could say that, oh, well, perhaps those who use hormone replacement therapy are more likely to be healthier, less likely to have central fat, maybe of higher socioeconomic status, higher education, better health outcomes in general. And this in actuality is true. However, this finding is consistent with meta-analysis of randomized controlled trials that looked at hormone replacement therapy administration on fat mass and found similar results. And so in addition to fat mass, we looked at lipids as well. And so I led a study that investigated the changes in lipids around menopause. And we see that postmenopausal women, again, have an unfavorable lipid profile, but it's not all as clear as this. For example, the high density lipoproteins that we see, there was no difference between pre and postmenopausal women. So whilst there were some unfavorable lipid profile that developed in postmenopausal women, this wasn't the case across all measures. And indeed, when we consider the contributions of age, it was quite challenging for the lipid systematic review, given that we didn't have sufficient longitudinal studies for meta-analyses. So we largely use cross-sectional studies, or solely use cross-sectional studies rather. And we found that much of the variance, or a large portion of the variance, was explained across most measures. So we did a sensitivity analysis to try to address this age menopause question. We categorized women who had age difference of less than 5 years to see what the difference was comparatively to the whole group results. And we found no difference in magnitude, direction, or significance of effects compared to initial estimates for HDL, LDL, and total cholesterol. So it could be suggestive of an effect of menopause, which is possible, but it could also be other factors, including group differences, given insufficient longitudinal studies were available. And it also sort of maps on to the changes that we see in the other systematic review around fat mass changes, given the correlation between lipids and fat mass is high. And so the real interest for me after quantifying these changes precisely about what was occurring physiologically around menopause and fat mass and lipids, was seeing what the contribution was of these changes to brain health. And so I conducted a study that looked at longitudinal changes in fat mass in the hippocampus. And what I think was important to consider in this research question was to consider baseline weight. An assumption that I had in terms of designing this study was that the changes that we see in fat mass are not comparable across weight domains. I think there's a difference in one BMI point increase from 20 to 21 to 25 to 26 to 30 to 31. And so I wanted some way to sort of conceptualize that within the grouping of the participants to ensure that they represented not only the change that we see over time in fat mass, but also their reference or baseline level of fat mass and that contribution to brain health. And so I used the UK Biobank study for this data for this study, and so that's large population cohort. This study used the neuroimaging cohort within there, so I had 20,000 participants, and we categorized participants based on whether they remain normal, stable, so at a normal level of fat mass across time points, where they increased from normal to overweight or obese, whether they decreased from overweight obese to normal, or they remained overweight obese stable. And the categorizations were based on established or standardized thresholds for these different measures. And again, I took the approach of looking at different measures like waist circumference, waist to hip ratio, and BMI. And so what we found was that, unsurprisingly, individuals with chronic overweight or obesity, they had significantly lower hippocampal volumes when compared with those who maintained a normal level of fat mass. So overweight and obesity, particularly chronic overweight and obesity, was detrimental to some degree, or it was associated with lower brain volumes in terms of the hippocampus. Now, what was really interesting was that individuals who were normal, within the normal range at follow-up assessment, but had a previous history of overweight or obesity as measured as baseline assessment. So I think that was approximately on average 7.5 years before. And those individuals also had lower hippocampal volumes. And so to us, that spoke to the messaging that the detrimental effects of overweight and obesity may extend beyond the duration of overweight and obesity itself. So even if someone appears to be healthy or within the normal range, if the chronicity of obesity is something to consider. And this makes a lot of sense when we, again, come back to the mechanisms and consider how obesity is linked with changes in the brain in terms of pro-inflammatory cytokines and also in terms of metabolic syndrome. It's a central component of categorizing clinically metabolic syndrome. And so just to conceptualize the changes in the hippocampus and the magnitudes of their effects, which I think are really, really important to do when discussing research, not just the significance of the findings or the direction of effects, but the magnitudes. Here's a plot I generated based off of 2 systematic reviews that looked at trajectory changes in the hippocampus over time for under 55, 55 to 70, 70 plus in Alzheimer's disease. And what we see is typically in this cohort, which were 45 to 70, the change of 1 and a bit % of hippocampal volume is equivalent to around about 1 to 3 years of aging. And so that's what we see here. And so these cardiometabolic factors are important in regards to considering the different measures that we use. And so what was interesting was the largest magnitude of effect that we saw within this study was for waist circumference. And it actually makes a lot of sense with this subsequent analyses that we did, where I looked at the relationship between BMI, waist circumference, and waist to hip ratio with total body fat percentage and visceral fat percentage as measured through DEXA, which is sort of the gold standard in sort of quantifying fat mass percentage, and others, bone mineral density and lean muscle mass and whatnot. And what we found is that for total body fat, BMI actually does a really good job of indexing total body fat. And waist circumference as well is comparatively okay. But when it comes to visceral fat, which is the one of most interest when we focus on the brain as well in terms of its relationship to brain health, waist circumference, by and large, performs over and above in regards to its representation of visceral fat, which makes sense. And further to this, when we looked at subgroup analyses for women and men, In women, they consistently reveal lower hippocampal volumes in all the groups that I described earlier. So obese stable, obese to normal, or normal to obese, compared to normal stable group for waist circumference. But the results weren't consistent for these other measures. And I think in part that speaks to the mechanisms that drive the associations and what these measures are actually measuring. So it possibly demonstrates the utility of waist circumference to measure visceral fat. And it may also reflect the changes after menopause. As I noted in the previous systematic review, there's increases in central fat, trunk fat over time. This could speak to the changes that we see in brain health. And so collaborators and I, in those I mentioned earlier in Lausanne, worked on quantifying the relationship between other cardiometabolic health, in addition to ones I've discussed thus far, in relation to white matter hyperintensities. And we see largely similar story emerge. Postmenopausal women have a poorer cardiometabolic profile than premenopausal women, and these factors are above an effect of aging, and these factors seem to be related to increased white matter hyperintensities, particularly for waist hip ratio, BMI, and blood pressure. We see this increase, but largely overall. And so the next focus that I wanted to look at was the relationship between menopause and the brain. And so I was curious to know, how do we sort of, if menopause is associated with all of these changes physiologically and biologically, what are their contributions that we see as measuring through menopause's relationship with brain health? And so again, I used the UK Biobank study to quantify the associations and to try to understand better the differences that may be attributable to menopause versus aging. And so what we found was that postmenopausal women experience a greater reduction in total brain volume than premenopausal women, which is in some ways apparent here in the plot on the left. You can see that slope is a greater reduction in postmenopausal women, which are the teal dots. And so that was about 0.23%, so 0.25% for every 1 year increase in age. And so for postmenopausal women, every 1 year delay in age of menopause after 45 was also associated with smaller brain volume and hippocampal volume. So we see this effect emerging where the greater the delay in age of menopause was associated with smaller and smaller brain volumes. And what was quite surprising to me was that postmenopausal women in the UK Biobank study actually had larger, when compared on group differences, had baseline larger total brain volume and hippocampal volumes than premenopausal women. And this effect drove me crazy. I looked at it in all different ways. Multiple regression models, of course, that adjusted for age, but also propensity matching, which exact matches participants on age, and nearest neighbor matched participants on other key covariates, and also age-restricted analyses for 45 to 55. We found consistent results across all of them that postmenopausal women had larger brain ages. And I think this is a good reminder of two things. One is the fact that of the sample, UK Biobank is a great resource, but in some ways there are nuances in the sampling that may not be representative of the broader population. But it also speaks to whether there's differences between those groups and also the within group variability. When we talk about these estimates that I'm presenting here, they're reflecting averages, as most estimates, all estimates from regression models do to some degree. And so we need to consider that level of variability that we see in these plots in some way can contribute to the results that we're observing. And so I thought it'd be interesting to present you with research as well that I've done this year in regards to the contribution, as I noted earlier, the sex and genetic interactions that we see in their contributions to brain health. And so one question of particular interest for me was, what was the relationship between hormone replacement therapy and brain health as measured by structural and cognitive assessments, and whether this depended on ApoE status of participants was one of the focus questions within this study. And we can see in this top line of the plot, which looks at cognitive variables, largely there was no difference that emerged. And it was a really interesting finding, not because of what it was itself, but in the context of the literature, because there were several studies that looked at this part of research in terms of the contribution of hormone replacement therapy to cognition and whether it depended on ApoE status, and indicated an interactive effect. So YAP here suggests there's an interaction between estrogen use and E4 presence with a p-value of 0.037. Salah states here that there's an interaction as well with these variables with a p-value of 0.009. And Kang seems to indicate that there was an interaction as well. But when you look at these papers in further detail, that's where an interesting finding emerges. So for example, in Yap's paper, after they adjust for factors including age and education, like the usual suspects to covary for in research relating to brain health, the interaction no longer was significant. But instead, it's discussed as the interaction remained and lessened somewhat in statistical significance. And the abstract shown earlier only reports the first unadjusted model there. In Salah's paper, when you look at the interaction effect of 0.009, if you go to the table that shows the interaction effect, you notice a little letter A next to the 0.009. The corresponding text beneath that table states insignificant after correcting for multiple comparisons. But this information isn't really anywhere in the figure that depicts the interaction, as you can see the interaction term here, here in the delayed memory index, nor is it found in the abstract as well. And then also in Kang's paper, the interaction actually doesn't meet the threshold for statistical significance and is instead discussed as a suggestive interaction. And I think this is really, really important and isn't isolated to these papers. It's sort of ubiquitous in scientific research in terms of how findings are discussed. And I think it's really important when it comes to science communication, and because what these findings say matter, and how it's interpreted can be largely influenced by what's said either in the papers or by the authors who write the papers. And so here's papers, here's articles that have come recently in the last few years in regards to hormone replacement therapy. It could reduce Alzheimer's risk, early study. Both of these studies were referencing that Salah paper, which showed it wasn't significant after correcting for multiple comparisons for that cognitive measure, and was only one item within the entire memory index as well. And so this is largely a motivation for myself to engage in science communication. I think it really matters in terms of getting the messaging out there of what the research is about. And so when I do research, I don't just finish with the publication. I engage with the media at whatever university I'm part of. Here it's ANU initially, and to the right at University of Canberra, to sort of get that messaging out across to the public, because an informed public is really, really important, and it's no wonder why people are confused when it comes to what the findings are when there's these discrepancies within the scientific literature itself. And so part of that science communication comes through podcasting as well. I mean, you don't often, you have to always be charitable to research that's presented in newspaper articles, because researchers don't often control the narrative as much as they'd like to. And in part, that's why I love radio interviews, because it allows for me to speak clearly and explain research findings to the public. But also I run a podcast as well, Midnight Conversations, and you can achieve impact through these things, which is through a microphone, and you're able to reach a large number of people. It has 55 episodes, over 7,000 plays, and it's on Spotify and Apple. And I think it speaks to the most common question I get asked when I tell people what I do, which is, what can I do? What can I do to help improve my brain health and help ensure that I don't get dementia or reduce my risk for dementia? And I think this is where it speaks to the importance of quantifying the magnitude of effects. What effects have the largest contribution to the changes that we see in brain health? And these Livingston articles do a really good job of starting to articulate that narrative of, well, what is the potentially modifiable contribution of risk factors, and of those, which one should we be directing our focus and attention towards? And here on the left, when you look at the mechanisms as well, you can see exercise quite centrally there has important roles in reducing brain damage, cognitive reserve, and also reduced brain inflammation. And so I think a big question for me is in regards to understanding and quantifying precisely the magnitude of these effects for brain health. And I think big data is certainly a path to being able to help answer that. Now, there's much more work to be done. And so I'll sort of leave you with this overview of the dementia predictions and the models that are currently in existence. And what I want to show you from this paper is they compared all of these different models, including ones like the ANU-ADRI, which are developed here in Canberra, the CADE, the UKB dementia risk scale, all sorts of dementia risk scores. And what they find is that when they model age alone, which is the green line here, you can see that not many of these risk scores are performing much better than age in predicting dementia. And that's unsurprising to some degree. Age is the biggest predictor of dementia. And the ones that perform the best are those that consider ApoE status. And so age and ApoE are two of the main contributors to dementia risk. And so we have to think about, well, how can we have so much more work to do to improve these models. And we need to consider things such as sex differences that emerge when we look at considering the contribution of these risk factors. And also statistically, many of these models, if not all, use frequentist statistics. And so considering a more Bayesian approach that considers prior knowledge of our understanding of these risk factors is important to improve, as I said at the start, the accuracy of these models and the utility of them for individuals. And so I guess sort of that comes full circle to the questions I raised at the start. Hopefully you get a sense of the interests I have and how these papers fit within the scope of those questions that motivate what I do. And if you're interested in collaborating or want to talk more, I'm always open for a chat and meeting. So feel free to let me know. Thank you for the invitation to come. And yeah, thank you.
Great talk, Anandan. Thank you.